So first of all, we are going to discuss today about proteins. We have discussed about the carbohydrates, one of the major portion of our diet. And second, we are going to see about the proteins. And third, we'll be discussing about the fat. Second important, that is the proteins. We are going to talk about the proteins in detail. This is the content that we are going to see. That is, first is we are going to discuss about the introduction, types of protein, amino acids, classification of amino acids, structural organization of proteins, amino acids and bases, peptide bond and hemoglobin and myoglobin. So these all things you'll be understanding in the basis. About carbohydrates, we have discussed about the introduction. Later on, we are going to see about what are the processes, that how we digest them, how it is absorbed, which are the processes involved in absorption and breakdown of a single molecule of glucose, and how energy is produced from it. So that is for later on. Now, this is an introduction to proteins. So proteins as a whole, you can say they are formed from the basic molecules of amino acid. Many amino acid group combine together to form one polypeptide or protein. Yes. So protein is also the basis from which the cells have everything. Cells ne communicate karvu hoy, energy produce karvi hoy, enzymes which are completely proteins, they have catalysis action. So these all are the processes. So you can say proteins are the building structural blocks of any information transmitted in or outside the cell. So we'll see the definition. So proteins are molecular machines or building blocks and arms of a living cell. Yes, a living cell needs some information to be transmitted within itself. Nucleus mathi, DNA mathi koi પણ એક enzyme બનાવવું હોય તો પણ protein will be responsible, and that enzyme or the protein that is formed through such a process એને act કરવું હોય તો પણ it is required that proteins have to work accordingly. Proteins are polymers. They are built up by amino acids that are linked to a peptide chain. So here you should remember NHCHCO group. Yes, this group is peculiar to amino acids. So NH group, hydrogen attached to carbon, and CO group along with one that is side chain. This is one amino acid NHCHCO along with what is the side chain. Side chain are various groups, that is, it may be negatively charged, positively charged, hydroxyl ions, neutral ions. What all ions are attached here will determine what type of protein it will be or what type of amino acid it will be. And accordingly a ek thyu એવું પાછું બીજું એવું ત્રીજું, and accordingly a long chain will be formed, hence the term polymers. Yes, polymers means multiple amino acids will form together, form a peptide bond, and because of this peptide bond there will be complete one protein molecule. But important is NHCHCO group, and along with that side chain will be attached. There are 20 main species of amino acid residues. Their position in the protein chain is gene encoded. Yes, you might have learnt about the DNA and RNA. Yes, so both of these are specifically formed from such proteins, from such amino acids. So, okay, what is a gene first of all? What do you understand by gene? Sorry, can you repeat? Wrong, sorry, didn't get it.
Sir, it is a characteristic which is carried from parent to offspring.
Okay, so it is something that is carried from parents to offsprings. Okay, fine. So what is the difference between DNA and a gene? It is important to know these terms specifically. See, DNA is the whole double helical structure that you have studied, what you can see here in the upper right corner of the slide. Yes, specific portion of the DNA, say for example this strand which is consisting of 4 amino acid, which will be termed as gene. Yes, specific portion of DNA is known as gene. Yes. So if you want to make any protein out of DNA, specific genes will be responsible, which will be decoded and encoded. Yes. You might remember the term tRNA and mRNA. Yes. Messenger RNA and transport RNA. So these were responsible for specific part of the DNA, specific part of the DNA લેશે એને ટ્રાન્સક્રિપ્ટ કરશે, ટ્રાન્સક્રિપ્ટ કરીને ટ્રાન્સપોર્ટ થશે, ટ્રાન્સપોર્ટ થઈને નવું પ્રોટીન બનશે. Yes. So it is written their position in the protein chain, this is the whole protein chain DNA, is gene encoded. So specific portion is responsible for specific gene. Yes, and accordingly new proteins will be formed from them. There are 20 in living human beings, there are 20 main types or species of amino acids. You do not need to remember all 20. If you can, that is well and good, but depending on the R, that is the side chain, they are divided. There are 5 main classes, which are there we are going to see, but accordingly you will have to remember any 1 or 2 example for each. The peptide bond, that is the bond formed between, so here you can see the new peptide formed bond that is formed between the two molecules of amino acid. So here you can see this is, you can say this is the peptide bond formed between the CO and NH. This is so flexible, it can rotate, it can be embedded into helical structures like this, it can be made into sheets, yes, like a paper sheet it can be made, and accordingly the structure is named. So the peptide bond allows for rotation of protein, therefore protein can fold and orient the R group in favorable position. You can say they are liquid type, they can be molded in various shapes. Yes, and accordingly R group can be changed, their position can be changed. So this is an example of how a peptide bond is formed. So you can see C and NH, CO and NH group, they are uniting, and this H2O will be removed. Yes, if H2O is removed, this is which type of reaction? Oxidation or reduction, or any type, other type of reaction? If OH group is removed, it is which type of reaction? Yes, oxidation, reduction, we have already discussed. ઓક્સિજન એડ થાય તો કયા ટાઈપનું રિએક્શન કહેવાય?
ઓક્સિડેશન.
ઓક્સિડેશન. અને હાઇડ્રોજન એડ થાય તો કયા ટાઈપનું રિએક્શન કહેવાય?
Reduction.
Good. So here if you see the reaction, hydrogen is being removed, so this will be termed as hydrolysis type of reaction. Why? H2O is being removed. This is not oxidation, this is not reduction, this is hydrolysis. Why? Because molecule of water is being removed, H2O. Yes, so this is hydrolysis type of reaction. We have discussed this already in the carbohydrates. Moving forward to types of proteins. So according to the environmental condition and general structure, protein can be roughly divided into 3 classes: fibrous, membrane and globular. Yes, fibrous means it is usually water deficit aggregate. Aggregate means cluster, means a group. Yes, so whenever they form a group, they will be water deficit, they do not contain water in them. Their structure is usually highly hydrogen bonded, highly regular and maintained mainly by interaction between various chains. What is hydrogen bond formed between? What is a hydrogen bond? What is it formed between? OH group mainly is hydrogen bonded. Yes, anywhere maximum efficacy of oxygen for hydrogen, otherwise this also is an hydrogen bond. Yes, and between this is termed as બે amino acid વચ્ચે કયું bond form થાય to form a protein? This bond is termed as we discussed it right here, peptide bond. Yes, So peptide bond, બે amino acid વચ્ચે bond form થશે, which is a strong bond. This will be peptide bond. And anywhere hydrogen is forming, bonding along with oxygen, nitrogen or carbon, that will be mainly hydrogen bond. Yes. So fibrous protein which are fibrous, it will be water deficit structure, usually highly hydrogen bonded, highly regular, mainly maintained by interaction between various chain. Membrane proteins resides in a water deficit membrane environment. Yes, our membranes, cell membranes, you know the cell membranes. Yes, it is semi permeable. Yes, I hope everyone knows what is permeable and semi permeable or non permeable. Semi permeable means selective amount of ions and proteins can pass through it. Yes, एक selective amount of ions and proteins જ એમાંથી pass થઈ શકે. Here membrane proteins are responsible for that. એટલે એ થોડાક જ એવા પ્રોટીન્સ છે અથવા આયન્સ છે જેને અલાઉ કરશે movement to and fro the inside and outside the cell. So these are the membrane potentials. It resides in a water deficit membrane environment. So here also water will not be there, it will be water deficit. The intramembrane portions are highly regular and highly hydrogen bonded but restricted in size by the membrane thickness. So you can say fibrous proteins and membrane proteins, they are both water deficits. But main important difference is membrane proteins, they are majorly present in intramembrane portions. Yes, within the membrane. Membrane ni બે layer hoy, ek outer layer, ek inner layer. Yes, outer layer will communicate with the outside of the cell. Inner layer will communicate with the within the cell. So within the cell જે પણ વધારે હશે proteins, that will be membrane proteins, and they will be responsible for allowing to and fro motion of ions and other proteins. They are again both mainly hydrogen bonded and regular. Yes, and size is restricted by the membrane thickness. So membrane proteins, their size is maintained or restricted by the membrane thickness. So membrane કેટલું thick છે, જો membrane વધારે thick હશે તો size of membrane proteins will be bigger. But if the membrane itself is thin, that means the size of the membrane proteins will also be smaller compared to other membrane proteins. And the last, that is the globular proteins. These are less regular. Structure is maintained by interaction of chain with itself and sometimes by chain interaction with cofactors. Cofactors, that is the side chain. So side chain કઈ રીતે react કરશે એની ઉપર આનું structure depend થશે, that is globular proteins. Yes, they are present in water also. They are not water deficit. First two, fibrous and membrane, are water deficit. Here it will be water rich. Yes, so water rich proteins are globular. Yes, globule form thau. Globule is like a sac. Yes, so it will contain some amount of water in them. So these are the three basic type: fibrous, membrane and globular. Fibrous proteins you mainly see in the cell and outside the cell. Membrane proteins you mainly see on the membrane, and globular proteins may be present within or outside the cell again, but the differentiating portion between fibrous and globular is presence and absence of water. So water deficit and water present, that is fibrous and globular proteins respectively. Amino acids, that is the building block. એક amino acid થી જ આખું protein chain બને. Yes, so amino acids are the basic structural constituents of naturally occurring proteins. They all consist of amino group. You can see we discussed about NH group. Yes, so NH3, carboxyl group COO minus, a hydrogen atom attached, so you can see the hydrogen atom, and a side chain bounded to a central alpha atom. So this is the alpha carbon also. There are 20 standard amino acids. These all things we have already discussed. So there is an amino group, NH group, carboxyl group, COO group, R group, that is a side chain, and one hydrogen ion attached to a alpha carbon. You need to remember this: amino group NH3 or NH if it deficits, બીજું bond form કરશે ત્યારે. Carboxyl group, that is COO minus, R group, that is a side chain, and H hydrogen attached to one alpha carbon, that is alpha C alpha. Yes, it is denoted by C alpha. હવે અહીંયા બીજું જ NH3 પાછું અહીંયા join થશે. There will be second alpha carbon, R group, H group and C double O group. Yes, so along with that C double O group and NH3 or NH group ની વચ્ચે જે bonding બનશે, that will be termed as a peptide bond. Yes, it is bonded, so it is peptide bond. They are now bonded, combined molecule. Yes, amino acid together form one large protein. There are 20 standard amino acids which will tell that which type of amino acid is responsible. Coming to the side chain, we were discussing now, there were 5 groups that we already told. I told you there are 5 groups and we will see examples of each. So amino acids are classified into 5 main classes based on the properties of the R group. Now if you see, amino acid ને classify કરવું હોય તો ખબર છે કે amino group constant છે, carboxyl group constant છે, hydrogen ion રહેવાનું જ છે, hydrogen bond through, and carbon ion તો fix જ છે. આટલો structure completely fixed છે. amino group, carboxyl group, alpha carbon and hydrogen. Change એક જ વસ્તુ થાય છે, side chain, that is R group. તો R group ઉપર જ આખું classification depend કરશે. હવે R group કયા કયા હોઈ શકે? તો first is non polar, there is no polarity, they are neutral. Second, aromatic. Third, they can be polar. Fourth, they can be positive, and fifth, they can be negative. Can anyone tell me what is polar and non-polar? What do you understand by polar and non-polar? Yes, please. Are you writing in the chat? I am not able to see simultaneously. Okay, what is the difference between polar and positive negative? See, first of all, you need to understand this. Non-polar and polar means presence of ions. Yes, plus and minus. Yes, non-polar means there is no presence of any ion. There is no positive, no negative. Barabar, neutral vastu hoy ne, you can say that is non-polar. And if any ion is there which is plus minus, but their sum is 0. Yes, so positive ions 3 છે તો negative ions પણ 3 છે, પણ એનું sum કરો એક molecule જે બનશે, that will be polar, but it will not be positive or negative. Yes, so polar, that is uncharged. Uncharged means there is some amount of charge, but the sum of which or total molecule is neutral, that is polar uncharged. Non-polar means there is no charge at all, no positive ion, no negative ion. Positively charged and negatively charged, so sum will be plus, so plus 5 and minus 3, so sum will be plus 2. Negatively charged, plus 5 plus minus 7, so minus 2 will be the negative charge. Depending on the charge, it is all divided. R group is divided on the charge, and accordingly it will be the amino acid. Amino acid કોના ઉપર depend કરશે? ખાલી R group or side chain. Yes, so side chain, they are 5: non-polar, aromatic, polar, positively charged and negatively charged. So there are 20 different types of amino acid. For each you need to remember at least one example. Yes, which are they we are going to see. So these are the non-polar or aliphatic R groups. So leucine, isoleucine and methionine. Yes, you might have heard such amino acids already in your 11th and 12th. You might have seen a table with all the 20, but no need to remember all 20. Remember example of one each of the R groups. Yes, non-polar, leucine, isoleucine, methionine. Second, aromatic, phenylalanine, tyrosine and tryptophan. They are aromatic. Why? Not because of smell, because of presence of such a benzene ring. Yes, you might remember the benzene ring that you might have studied in your chemistry. Yes, in your 11th and 12th you might have seen. So presence of such is termed as aromatic R group. So phenylaniline, tyrosine, tryptophan. Yes, this is an example of aromatic R group. Third, polar, that is uncharged. Yes, asparagine and glutamine. Yes, asparagine and glutamine. These are also the terms, or these are also the amino acids used in a DNA. Yes, which are part of the DNA. Positively charged, lysine, arginine, and histidine. Lysine, arginine, histidine. Yes, no need to remember the structure also, just remember the example, one or two example of each. And negatively charged, aspartate and glutamate. Yes, aspartate and glutamate. So these are the examples, 5 groups. I'll be sharing the presentation with you later, so don't worry, you can read it afterwards also and go through it. Remember one name each, one example of each. Moving forward, we'll discuss about the structural organization of the proteins. So first, there are 4 main structural organizations. Structural and functional feature of protein and protein complex addressed in the 4-level hierarchical organization. So first of all, when you see a protein, a complex structure, it will look something like this. Yes. Afterwards, if you see a small portion of it, if you break it down, it will look something like this. Again, if you focus on very small portion of this also, it will be something like this, and last it will be something like this. So this is the primary structure, the basic structure. એટલે કે ઘણા બધા amino acid એક લાઈનમાં ગોઠવાઈ જાય એને કહેવાશે primary structure. You can see lysine, glycine, and also valine, alanine, and histamine. So all this will be termed, all this are arranged in a specific order. When this specific order, as we discussed, peptide bond are highly flexible, ekdam flexible hoy koi પણ structure લઈ શકે, so they will form such helical structures. This is termed as alpha helix. Yes, e alpha helix પાછા ભેગા થશે and that will form peptide chain. This polypeptide chain, again, if done together, you will see a single amount of protein. Yes, so this gets complex every time. So first is the primary structure, 1 degree, that is also read as primary, secondary structure, tertiary, and quaternary. So it all starts with simple association or binding of various amino acids. They will form a special structure because the peptide bond is highly movable or configurable. Third, this alpha helix structure move together to form a polypeptide chain, and last it will be assembled subunit or a single protein that will give you a quaternary structure. Yes, એટલે simple છે. This is the simple form, this gets complex, and this is the last what you see in the cells or in the microscope of while examining proteins. So primary structure, it is a description of all the covalent bonds, mainly peptide bonds and disulfide bonds, linking amino acid residues in the polypeptide chain. So it is the primary structure. So you can see it is the peptide bonds in between and nothing else, formation of a simple structural unit. Most important element of primary structure is the sequence of amino acid residues. Because this structure, because of this series, જે પણ આ series form થાય છે, lysine પછી અહીંયા valine આવી જાય, valine આવી જાય, then it will form a different amino acid, when it will form a different protein. Yes, it all depends on the sequence કે કઈ રીતે આ unite થાય છે amino acids. Secondary structures refers to particularly stable rearrangement of amino acid residues giving rise to recurring structural patterns. So you can see this is recurrence patterns. You will see this one. Yes, one alpha helix, આવા ઘણા બધા alpha helix હશે. એટલે આ એક જ chain, this one type of chain itself will be responsible for forming many alpha helix. ઘણા બધા alpha helix બનશે and they will unite together to form the tertiary structure. So secondary structure, they are giving rise to recurring structural patterns. Tertiary describe all aspect of the 3-dimensional folding of a polypeptide. So this is, you can see 3-dimensional. Up till here, this is single dimensional, this is 2-dimensional. Yes, you can form a helix on a paper, that is 2-dimensional, but you cannot form this type of structure on a paper. This is a 3D model, you can see. So this is again a 3D structure. And quaternary structure, when a protein has 2 or more polypeptide subunits. So these are the polypeptide units. Again, they two will unite together and form a complete quaternary structure. Their arrangement space is referred to as quaternary structure. It will always be a 3D model. 3D model be bhega karichi, so it will always be a 3D model. So primary structure, normal single covalent bonding and peptide bonding in a specific sequence. एक specific sequence માં arrange થશે. Second, this specific arrangement can be molded in structures and this can be done recurrently. વારે ઘડીએ કરી શકાય. This 2D structures, alpha helix many, get together and form a 3D structure that is tertiary. Yes, and two or more tertiary structures or subunits unite to form a quaternary structure. Yes, so this is how a normal protein is formed from the amino acid. It all depends on the bonding, it depends on the structures that we are going to see. We are going to discuss about the alpha helix and beta sheet also, how they are important. Just few lines we'll discuss about them, but probably in the next lecture. For now, up till here, any doubts anyone has? So we discussed about what is a protein first of all. So NH group, CO group, that is utmost important. And why NH2, NH3 is not there and COO is not there? Because this is forming bonding. So H2O is already removed. So 2H and 1O from here, 1O here, 2H from here. They both are already removed. And the basic structure tells you there's always ammonia, NH3, COO minus carboxylic group, and hydrogen ion will be attached to the main alpha carbon along with the side chain. Depending on the side chain, there will be types of amino acids. Amino acid form the basic building block and arms of a living cell. Any information you want to pass between the cells or outside the cell, you always need a protein. Yes, proteins are made from peptide chain between various amino acids. Then we discussed about the sequence, how the peptide bond. So peptide bond also allows for rotation of protein. Therefore, it can orient the R group in favorable position wherever required. We discussed about the types of proteins. First of all, fibrous, membrane, globular. Fibrous and membrane, they are highly regular. They are maintained with hydrogen bond and they do not contain water. Membrane proteins mainly specifically present on the inner aspect of the membranes, and depending on the size of the membrane, their thickness is determined. Globular proteins will have water in them. That is the main difference. Later on, we described about the same thing: NH3, COO carboxyl group, amino group, R group, that is the side chain, alpha carbon attached to hydrogen, and depending on the R group, non-polar, without any charge, aromatic, presence of benzene ring, polar, that is charges are there but it is neutral, positive, positively charged, negatively charged, and these are the examples: non-polar, leucine, isoleucine, methionine; aromatic, phenylalanine, tyrosine and tryptophan; polar, asparagine, glutamine; positively charged, lysine, arginine and histidine; and negatively charged, aspartate and glutamate. Yes, so this was the example. And the structure: primary, mainly formed by just a sequence; secondary, forming a 2D structure, helical structure, alpha helix, which are reproducible, can be made again and again. વારે ઘડીએ બનાવી શકાય. ઈ પાછા ભેગા form a tertiary structure. Tertiary structure is a 3D one. The 3D one, when it unites two or more, it will give quaternary structure, that is complete the whole protein. Yes, any doubts till now?